And excitation contraction coupling is really the process of regulating muscle contraction. And this is where we think about what the calcium's role is in turning the contraction on and off. All right. So the sequence of events begins with what we spoke about at the neuromuscular junction. It's going to be dependent on the neural input by a motor neuron. That's going to require calcium to be released from the sarcoplasmic reticulum. So the motor neuron, where we have the neuromuscular junction, think back to where we had the highly folded end plate. We had the dense amount of acetylcholine receptors to bind that acetylcholine. Remember, the only neurotransmitter at the neuromuscular junction is acetylcholine. And the only type of receptor is going to be the nicotinic cholinergic receptor. So when acetylcholine is released from these motor neurons, it will cross the end plate, the motor end plate, and bind to those receptors, and that will bring about a specialized type of contraction on the motor end plate, which is called the end plate potential. So this is always excitatory. This always results, as long as there is neurotransmitter release, this is always going to result in excitation or contraction of the muscle because of the steps that we're going to look at here. So we spoke about the structure of actin, the structure of the regulatory proteins, and that is a big part of regulating. So if there's no calcium that leaves the SR, remember calcium is being stored in the sarcoplasmic reticulum, and then on the trigger of the end plate potential, that calcium gets released, and then it can engage with troponin, and it can cover up the, open up, or excuse me, expose the binding sites by moving tropomyosin away from those sites. So if there's no calcium present, troponin keeps tropomyosin covering up those myosin binding sites. Therefore, there's no cross-bridge cycling, so there's no sliding filament model happening. The muscle remains relaxed in that state. Therefore, no calcium means relaxed muscle. Alternatively, when the end plate potential arises, then calcium is released from the SR, then it can bind to troponin, then it can shift tropomyosin to expose those binding sites, and then we can have cross-bridge cycling. And so the presence of calcium means muscle contraction. All right, so this is exactly what we mean when we say the excitation contraction coupling. How are the events of excitation at the neuromuscular junction coupled to the events that actually lead to muscle contraction or shortening of the sarcomere? So let's look at those steps in sequence, and then we'll describe them in the illustration coming up. So the action potential gets to the sarcolemma. And remember, the sarcolemma is the fancy term for the muscle membrane, the highly folded motor end plate or the muscle membrane, similar to the cytoplasm. So when we think about the action potential arriving there, traveling down the T-tubules, remember the T-tubules being the invaginations along the muscle, and then that triggers the DHP and ryanodine receptors. The DHP receptor is going to open up a calcium channel, which is also coupled to that receptor. And this is where the ryanodine receptor is also acting as a channel. So these two work together to result in calcium being released from the lateral sacs of the sarcoplasmic reticulum. So now this calcium can flood the cytosol, and then it can regulate the actin filaments, and then it can result in cross-bridge cycling. Okay, so let's reiterate those sequence of steps here. And again, this begins where we left off at the motor end plate. So action potential arrives here at the motor neuron, the axon terminal, acetylcholine released across the junction, binding to these nicotinic receptors on the motor end plate. And so these are going to be able to allow sodium and potassium, more sodium, so that automatically results in an excitation or the end plate potential. The end plate potential will travel down the length of the sarcolemma or the muscle membrane. And then because of these tunnels, these invaginations, the T-tubules, the action potential can actually travel deep throughout the muscle as well, not just along the surface. It can actually burrow into the depth of the muscle fiber and go deep into the muscle as well. All right, remember, these are bundles of fibers that are stacked together. So in order for this end plate potential to be spread throughout the muscle quickly, it has to be able to go through the depth, and that is where the T tubules come in. The other function of the T tubules is to couple the action potential to the release of calcium from the sarcoplasmic reticulum. So the blue spaces here represent the lateral sacs that are formed by the SR that create this triad structure. So a lateral sac on this side, a T tubule in the middle, and then a lateral sac on this side together creates a triad. And so that coupling allows the voltage change that arrives on the membrane to stimulate the receptors that open up these calcium channels so calcium can leave the SR and make its way out into the actual sarcoplasm. Okay, so that is the importance of the T-tubule, the triad, and then how calcium gets released. Now calcium can bind to troponin, which is the pink regulatory protein here. And when that troponin binds calcium, it will expose the covered up binding sites. So these black dots are the binding sites for myosin. And so now the purple protein, which is the thread-like protein tropomyosin, will be dragged off of those binding sites. And as you can see here, now they are exposed. So now they can bind the myosin heads, and then they can be dragged towards the center of the sarcomere, and then we can see the muscle contraction, right? As the sarcomere shortens, the muscle fiber shortens, the entire muscle shortens and contracts. Now, in order for us to cease muscle contraction, we have to be able to turn this off. So this is why it's important to think about calcium, the presence of calcium turning the contraction on, but the absence or removal of calcium from the sarcoplasm and putting it back into the SR is extremely important for turning the contractions off, for allowing the muscle to go back to its relaxed state. All right. So that is now going to happen by the way of active calcium pumps, calcium ATPase pumps that pump this calcium back into the SR. And then now we're going to see it's no longer binding to troponin. So then troponin will then cover up those binding sites with tropomyosin, and then the muscle will go back to a relaxed state as cross-bridge cycling will cease. So calcium's presence turns the contraction on, and then calcium's absence turns that contraction back off. So this is the state of the actin filament. Here's the troponin complex. Notice it has 3 different binding spots, or 3 different units. 1 to bind the actin at the bottom, 1 to bind the troponin in the middle, the tropomyosin, in the middle, and then 1 at the end to bind the calcium. So in the relaxed state, it's covering up, tropomyosin that is covering up the binding sites. And then in the contracted state with calcium present, it binds to its spot on troponin, which causes it to drag the tropomyosin fibers off of the binding sites. And now the binding sites are exposed so that myosin can engage. So that is how it becomes contracted when myosin can bind. All right. Now I've put these videos, I think they're really helpful at illustrating the process of this regulation, how contractions start by way of the events at the neuromuscular junction. So definitely look at these. I've linked them in Brightspace as well, but they're also in the PowerPoint. There's a part 1 earlier, and then there's part 2 here. Now let's look at some of the details of that T-tubule, that triad structure. So the 2 receptors that we mentioned, the DHP receptor and the ryanodine receptor, let's look at how they're actually gating the sarcoplasmic reticulum. It's sort of a gating mechanism that's happening here. So here is the sarcoplasmic reticulum, the SR in blue. And then here is the sarcoplasm, which is where the calcium needs to be in order to engage with the actin and myosin filaments. So these receptors are going to be voltage gated dependent. That is, when the action potential travels in between the T-tubule, it's going to trigger the DHP receptor, which is a voltage sensor. So when the voltage changes, it will trigger the ryanodine receptor, which is tightly coupled to open up the calcium channel. So this is going to be called calcium-induced opening, as well as calcium-induced closing, which means that whenever there's an excess of calcium, that is going to trigger the opposite event, and that is actually going to close these ryanodine receptors. So calcium-induced opening, meaning that calcium flooding this area because of the voltage change, which triggers the DHP first, and then triggers the ryanodine, which is also a channel. So instead of calcium being in the SR, it can now leave the SR through this channel and make its way into this space, which is the sarcoplasm. And then now when we have too much calcium flooding the SR, and we're sort of maxing out all of the filaments so that the muscle is at its maximum contraction, then that is also going to trigger these ryanodine receptors to close, and that's going to be called calcium-induced closing. So the more calcium will trigger more opening, but to a point where excess calcium, to where all of the filaments are saturated, actually triggers a closing of these receptors. Okay. And then also just thinking here about what we mentioned earlier, the termination of the contraction, putting the muscle back to its relaxed state. That is reliant upon calcium leaving troponin. So calcium moving away from the troponin binding site, which is that pink regulatory protein. And then that is what allows the calcium ATPase pumps to actively pump that calcium back into the SR, and then the muscle can go back to its relaxed state. Okay, so that is extremely important in understanding how we turn the contraction off and how we put that muscle back to its relaxed state. Okay. All right, so I'm going to end here just because I won't be able to get very far into the next learning objective. But our third learning objective is where we're going to get into the mechanisms of skeletal muscle contraction.